Good afternoon, everyone. My name is Ayodeji Ogunbayo. My talk is on the application of next generation sequencing and metagenomics in deciphering the human microbiome, and I'll be focusing on respiratory virome. I'm from the University of the Free State, Next Generation Sequencing Unit, and Division of Virology, South Africa. I'd like to say a very big thank you to the organizer of this symposium for giving me this opportunity to partake in this presentation. So here is the outline of the talk, and the goal of my talk is actually to broaden our horizon on the language of virome and that of metagenomics. As we know, the microbiome is defined as the collective genomes of microbes that lives inside and on the human body. It is believed that over 1,000 different species at least live in and on the body. So we may say that we have at least about 10 times as many microbial cells as human cells. So what is the human virome? Human virome is a virological part of the microbiome. It's a collection of all viruses that are found on humans. And it is a highly diverse system, and each body compartment harbors distinct viral population, from your skin to the gut to urine. More so, parasites they were believed to be sterile, they are clearly not sterile. Urine is not sterile, eyes is not, and even the lungs are not sterile. So what does the human virome contain? It contains your eukaryotic variants ranging from your DNA to RNA viruses. Some are pathogenic, some are causes asymptomatic infection, and the role of some is still currently unknown. So the sources could be from environment, plant, food, or even animal exposure. The virome also contains the bacteriophages. Some are the modulators of the human microbiome, some are reservoirs of functional genes, and some interact with the immune system. It also contains your endogenous retrovirus, some that are permanently fixed, the proviruses that are stably integrated, and even episomes that may induce dynamic infections. So what are the components of the human virome? These circles here, right here, represent the components of the human virome that can be characterized through metagenomic sequencing. And right here are the questions and implications that are associated with each component that can be answered. For instance, if we characterize the components of pathologies of unknown origin, we may be able to identify a possible viral pathogen. If we characterize the components of novel virus, we may be able to associate it with a disease, even perform virus characterization, or even epidemiological studies, amongst other components that can still be characterized. So the question is, why respiratory virome and respiratory tract viruses? We know that human respiratory tract is heavily exposed to microorganisms. And it serves as a very good baseline when performing viral analysis. And of interest is that respiratory tract infection, they are a significant cause of acute illnesses, and they are often associated with extensive morbidity and mortality, especially in children that are less than 5 years of age. And even with advanced diagnostic tools, in a lot of studies, the virus can only be detected in about only 40% to 80% of respiratory tract infections, meaning that the causative agent is either an emerging respiratory viruses or a possible virus with unreported mutation. We've seen such cases in our unit here, whereby about 10 out of 84 children tested negative. Those children were actually admitted for severe acute respiratory infection, and they tested negative for 18 viral pathogens and even 3 bacterial pathogens. So what are the approaches to sampling the human respiratory virome? We have the non-invasive approach, the less invasive approach, and the more invasive approach of using bronchoalveolar lavage. However, we often go for the use of nasopharyngeal swab or oropharyngeal swab or nasopharyngeal aspirate. In that, this less invasive approach is able to give us the idea of the spectrum of viruses that colonize both the upper part and the lower part of the respiratory tract. So viral metagenomics, we know viral metagenomics, what does the question that it aims to answer? It aims to answer how many viruses are present. We're talking about the diversity. So what are they? This answers the question of their taxonomy and also their function of biology. What are they doing there? So viral metagenomics, there are a number of methods involved in viral metagenomics, and some of them are listed here, from enrichment down to bioinformatic analysis. However, with enrichment, depending on what your end game really is, are you looking for DNA viruses or RNA viruses? Depending on that, you decide on your level of centrifugation, applicability of filtration, nucleus treatment, and even ribosomal RNA removal. We also have the part of RNA extraction. And mind you, you need to understand that the choice of RNA extraction has a very significant impact on your viral recovery. That should be at the back of our mind all the time. And we have the issue of random amplification, which is still currently debatable before we go to the DNA amplification. And then we have our library preparation, the sequencing, depending on which sequencer you're using, down to the bioinformatics aspect. You may use your alignment-based analysis or maybe de novo assembly. So the question is, what is the need? Virome study is still in its relative infancy, especially in Africa. Currently in Africa, there are limited data on the spectrum of viruses in children, both in health and disease. And we need to be transformative. We need to turn the science of emerging viral disease into a big data science. It's time for us to start making the unknown known. And this will result in converting virology into a data-rich field. So what are the questions that are yet to be answered when it comes to respiratory virome or respiratory viruses? Some of my favorite questions that are yet to be answered include, is there an interplay between virome and immunity? And also the beneficial effect of some certain viruses that are present in the respiratory tract. Other questions include any possible for novel viral discovery or even detection of viruses previously unknown tropism. So there are challenges in viral metagenomics. As we all know, all those are very much inevitable. We have the inherent need for an ample amount of certain nucleic acid. We also have, especially RNA viruses, they are always present at low abundance and they may be lost during enrichment. We also have a very big challenge of contamination with bacterial DNA. And even after having your results, you could still have a challenge of lack of read matches against databases. But currently, what have we done? In our units at the UFS NGS unit here, we tried to optimize the protocol for the detection of respiratory RNA viruses. We did evaluate different extraction methods for the optimal recovery of RNA viruses from respiratory samples. And also for the enrichment parts, we evaluated the effect of DNase treatment only and a combination of DNase treatment and RNA removal. So based on our in-house developed protocol, we successfully performed viral metagenomics on respiratory samples from children that are having severe acute respiratory infection and those without severe acute respiratory infection during winter and summer period of 2020 and 2021. We performed the patient recruitment at the tertiary hospital here, and that was after all ethical considerations were observed and approvals were granted. About 150 children with SARI, as per definition of World Health Organization, were recruited, and children with non-SARI, these are children that were admitted based on other conditions, but nothing to do with the respiratory infection as at the time of sample collection. They were also recruited. And all these children were under 5 years of age. The nasopharyngeal and oropharyngeal swab were collected per child in a viral transport media, and this was sent to the NGS unit for metagenomic sequencing. The point is that we performed this metagenomic sequencing in pools, both for SARI and non-SARI group. These pools were done in pools of 10 to 20 per pools. So, as you can see here, this is our preliminary results for now. So the metagenomic analysis, this is for the metagenomic analysis in children with SARI. We can see that both in winter and summer, these children with SARI presented with a highly diverse population of viruses. Their expression of viruses cohabiting these children are very diverse, although mostly they consist of Picanoviridae, Totiviridae, Ricoviridae, and Pneumoviridae family. And when we look at the colonization of children without SARI, both in winter and summer, we can see the less diversity, they're less diverse compared to those children with SARI. And they are mostly colonized by the Picanoviridae and Ricoviridae family. Our result actually corroborates other results that has been performed outside of Africa as well, and that has been published. So in summary, we are beginning to define the scope of the human respiratory tract virome. Although detected viruses in these children may not directly cause respiratory illness, but their presence may still impact the trajectory of illness through mechanisms that we still do not understand yet. More so, we still require methodological improvement when it comes to studying the human respiratory virome. Of note and of interest is that during this period of study, no Automyxoviridae family was detected during this period, which also corroborates the published results that on the disappearance of influenza viral family during the COVID-19 pandemic. So what next moving forward from this? We need the analysis of larger sample size. We need possible research on alteration of virome for health promotion. And we also should be able to associate newly discovered viruses with disease. And also, more importantly, we need extensive collaboration with immunologists and bioinformaticians. I would like to thank the immense support from our funders, Bill and Melinda Gates Foundation, William Emeritus Research Fund, and National Research Foundation. I also like to appreciate the immense support of my promoter and guidance, Professor Martin Inyanga, and that of my colleague at the university here. Thank you.
Thank you so much for your nice presentation, Emmanuel. So I would like to invite the participants to ask questions.
I have a question, and that is, if you could comment on what are the main challenges of working and doing molecular biology in your settings?
Okay, the main challenges relating to what actually?
I mean, reagents or electricity. What are the challenges, the problems that you face that make your work hard when you are in South Africa?
Oh, currently I cannot say there's a basic challenge regarding to maybe reagents, electricity. No, nothing of such. I think the only challenge I could say maybe if, and that's if we have a challenge with probably reagents maybe not working, or maybe getting the protocol to work. Maybe, yeah, maybe basically that. And that's an in-house thing. Everybody needs to develop their protocol and get it to work. So beside that, I don't think there's any significant challenge we experience.
Thank you.
Thank you. I think maybe I throw one question. So John asks you that, do you expect any difference in human virome with the presence of coronavirus?
Yeah, we do expect, because there's this recent publication that says when a child is co-infected with coronavirus, there are more chances of high level of co-infection in children that present with SARS-CoV-2 more than children that present with other kind of viruses. For instance, in our studies, at least about let's say 80% of children that they were having SARS-CoV-2 also had other viruses co-infected. And other children that were probably having, let's say, RSV only, or let's say rhinovirus only, the chance of them being co-infected is very minimal.